The overall goal of this procedure is to collect hearts by rapid manual dissection from live zebrafish embryos to obtain cardiac-specific mRNA. This is accomplished by first pipetting many anesthetized embryos up and down through a narrow pipette tip to simultaneously release the hearts from the embryonic bodies. Next, the dissected hearts are separated from embryonic debris in 2 rapid filtration steps. Then, the fluorescently labeled hearts are manually sorted from remaining debris. Finally, cardiac-specific RNA is extracted from collected hearts. Ultimately, the heart dissection protocol is used to collect sufficient amounts of embryonic hearts to perform transcriptome analysis.
This method will help to ask key questions in the zebrafish heart developmental field, such as which transcripts are expressed within the heart at different developmental stages, and also compare wild-type and mutant heart expressions.
To obtain zebrafish embryos for heart extraction, cross adult transgenic fish expressing a fluorescent protein in the heart, such as transgenic MYL7 EGFP. Under the stereo microscope, monitor the development of the embryos. Verify that the hearts are indeed fluorescently labeled and manually decorionate the embryos. When the embryos have reached the desired stage, transfer around 1